Today's presentation will be on genetic toxicology. My name is Leslie Recio. I'm the VP of Research and Development at ILS. Genetic toxicology is expertise in toxicology where we use a number of biological systems to assess whether a chemical can cause DNA damage in bacteria, in somatic cells, or in germ cells. We use, as I mentioned, bacterial cells and mammalian cells in culture and on animal models. And we use animal models in basically the last stage of genetic toxicology testing, since they incorporate absorption, distribution, metabolism, and excretion, which we cannot mimic in an in vitro system. Our test systems are designed to assess DNA damage in bacterial, somatic, or germ cells. And when it happens, when you damage cellular DNA, that's referred to as genotoxicity. When you damage cellular DNA, the genome responds. This is some of the newer revelations about how cells respond to environmental stressors. In the case of DNA damage, there's a well-described DNA damage response that's regulated by the gene P53. If there is some DNA damage with replication, you do have that potential to pass that bad DNA or bad information on to the progenitor cells and doing replication, and this could result in a mutant somatic cell or a mutant germ cell. So genetic toxicology is basically focused in two areas. One is regulatory genetic toxicology, and this is the type of services ILS provides to its clients, where we conduct OECD guidance genetic toxicology tests. This is a genetic toxicology test battery, which is called ICH-S2R1. That stands for International Council for Harmonization. And this is the genetic toxicology battery that was put forth in 2011. The US EPA also has its genetic toxicology test battery, which is very similar to the ICH-S2R1. And there are a number of international environmental requirements for testing. And for instance, REACH in Europe is known for one of the regulatory genetic toxicology as well. On the investigative side and research side, a lot of the research in genotox is focused on DNA repair, oxidative stress, the whole world of endogenous mutagens, where we have built into our diet exposure to known genotoxic compounds, carcinogens. There's also a risk assessment area, which is area ILS is focused on, where we use quantitative studies to develop a benchmark dose, high throughput systems, genotoxicity assessment in metabolic competent human cells. This is another area where ILS is involved in, and toxicogenomics. And that's basically to assess mode of action, assessing whether a compound has a genotoxic mode of action or a non-genotoxic mode of action, which is very important for risk assessments. And it makes a huge difference in how a compound's, the risk of a certain compound or exposure is assessed. This usually involves dose response and time course studies, gene expression profiling, so-called transcriptomics, and in ILS, we're using state-of-the-art next-generation sequencing to derive gene expression data for use in risk assessments. Before I get into some of the studies, I need to get into some basic biology. One is the mammalian cell cycle, and this is shown on this slide. When we look at chromosomes if you look at pictures of chromosomes, that happens in a very narrow window of time, basically during the mitosis, where the chromosomes are aligned, and this is metaphase chromosomes. The rest of the time, chromosomes are all unwound, basically like a bowl of spaghetti, and this occurs as part of the cell cycle as cells go through the cell cycle. The cell cycle stages are called G1, S, and G2. G stands for gap, and S is DNA synthesis, so it's gap 1 and gap 2 prior to DNA synthesis. Those times in there are the times it takes for a CHO cell to divide. So you can see that mitosis is a very narrow window of time, half an hour, and to get cells to be able to look at them microscopically for chromosome damage, we add cytochalasin and other compounds that inhibit cytokinesis, so you can actually capture the chromosomes during mitosis and capture them on metaphase. These are used to assess chromosome aberrations in Chinese hamster cells and human peripheral blood cells. One reason we use Chinese hamster cells is they have 22 chromosomes, and they're very big, and it's easy to see when you have a chromosome aberration when someone is scoring this by microscopy. By the way, scoring these kind of chromosome aberrations, it takes expertise and takes someone sitting down and actually looking at the cell and counting the chromosomes, and it takes a long time to do that. So how is DNA damage initiated? Well, it's initiated by either the compound itself is electrophilic or it's metabolized by our bodies into electrophilic compounds, which can then bind to or interact with DNA. DNA is nucleophilic, so if you have electrophilic compounds or you generate electrophilic compounds, they can bind to DNA and cause what we refer to as DNA adducts or DNA lesions. Well, compounds can bind to DNA at a number of sites. On the left-hand side are the base alkylation sites, where a compound can theoretically bind to the bases of DNA. One of the more prevalent positions of binding is N7-guanine, and that's shown here on the outside here. And N7-guanine, however, is the most commonly alkylated site, or actually there's a lot more methods to develop to look at N7-guanine. However, it's referred to as inconsequential because guanine, N7-guanine, does not participate in hydrogen bonding with cytosine. The hydrogen bonding occurs to these moieties here with cytosine. So if you don't in contrast, if you look at O6-methylguanine, ethylguanine, induced by ethylmethane sulfonate, you'll see that you get a binding at O6 of guanine. That O6 is involved in base pairing. So now you have a situation, we have an alkylation at one of the base pairs that forms the hydrogen bonds, and that disrupts that hydrogen bonding, and that is referred to as a pro-mutagenic lesion. The N7-guanine is primarily used as a dosimetric. We have compound-bound DNA, but if you want to ask the question, is that N7-guanine referred to as a pro-mutagenic lesion? It's not. Typically something like O6-guanine, which disrupts hydrogen bonding between guanine and cytosine, is considered a pro-mutagenic adduct. And it makes a big difference on what you're measuring, what the dose is. However, DNA is continually under attack. We have a number of lesions that occur in the DNA endogenously, apurinic sites, which basically means you lose a purine base. You have adducts formed from environmental exposures, breaks, and all these occur endogenously. And this has actually been measured by a fellow by the name of Jim Swinburne at UNC, where they actually quantified how many of these lesions you have per day and per cell. And so if you look at a cell, you have something like 30,000 lesions per cell of these various different types of DNA adducts that are formed. For instance, you have formaldehyde adducts. You also have acrylamide adducts in your body from your diet. And some of these adducts are like the ethenodeoxyguanosine, that second to last one, is the same adduct that's formed by vinyl chloride, which is a well-known human carcinogen. That occurs in our body endogenously. So you constantly have DNA damage in your body. They've formed lesions in your body, and some of these are identical to what you would see if you were exposed to a compound like, for instance, vinyl chloride. So what are the types of genotoxic mutagenic agents? Some of the most common ones that we are exposed to are endogenous mutagens, from our diet, polycyclic aromatic hydrocarbons when you grill food, and also acrylamide, which is basically a product of starch, incomplete degradation to form acrylamide from some amino acids. And there are also some physiological processes where, for instance, you have an inflammatory response. Macrophages come in, they release reactive oxygen and other deadly molecules to kill the invaders, but it's also potentially causing endogenous DNA damage. Chemical mutagens, the most common one are cigarette smoke and air pollutants. Some of these direct-acting agents, EMS and ENU, these are alkylating agents. These are typically used as positive controls, for instance, when we run our assays for genotoxicity here. And there's a whole number of other ones. One of the more prevalent one that's been studied quite a bit is, for instance, benzoypyramine. But some of these other compounds, topoisomerase inhibitors, spindle poisons, many of these are cancer chemotherapeutic agents. So many cancer chemotherapeutic agents are genotoxic, and that's how they kill cells, by breaking the DNA, breaking the chromosomes, causing cross-links, and that kills the rapidly dividing tumor cells. There are also a number of naturally occurring substances which are genotoxic, aflatoxin, physical mutagens, and also biological mutagens such as viral infections which insert into your genome and cause a DNA damage. So what is considered to be a mutagenic compound? Well, it tends to be a compound that can either react with DNA and causing mutations. So mutagens produce positive results in a number of test systems. We have developed a number of test systems to detect these kinds of damages, and we can measure this both in vitro and in vivo. So chromosomal damage is one of the first items that we'll talk about here, it's a measure of it's a measure of genotoxicity and potential mutagenicity. In this particular case, cells are examined for chromosome aberrations, and again, you think about the cell cycle, we're looking at mitotic cells that are basically at metaphase, and we're looking at those chromosomes. We measure chromosomal damage in the first cell pass to mitosis. So these cells really aren't going through cell division. They're just going only basically to the point where you have cells in mitosis, and they're not completing the cell cycle, forming two daughter cells. And that's what we do. We do that to be able to capture the chromosomes at that point and be able to score them. And right now, the guidelines require scoring of 300 metaphases per sample. So someone has to sit down and look at per animal, 300 metaphases, and score them for chromosome aberrations, chromosome deletions, or all the types of aberrations that can be detected using these test systems. This is quite different from a mutation assay. Mutation assays require cell division and require fixation of that mutation. So this shows a schematic, the difference between a test like chromosome aberration, which is a measure of genotoxicity, compared to a measure of mutation. And mutations are heritable. So when we do our experiments here with mutations, either the Ames test or in vitro mammalian cell TK assay, cells are exposed. That exposure causes a DNA adduct, add guanine, for instance. However, that's not a mutation. That's an adduct formed in the DNA. For a mutation, is a heritable change. So that cell either repairs that damage or that cell replicates now, and you now have a situation since that G is altered, it will not pair with C, it'll pair now with T. So now you have a G to T in that position. That's still not a mutation, it's a heteroduplex DNA. So that requires yet another round replication where now the daughter cell goes to the G and C, now you have an A and T, so you have a daughter cell, which now in that two daughter cells in this position, one has a GC, the normal base pairs, and the other one has an AT. And so this is called mutation fixing, and it takes clonal expansion. After that time, we have to wait some time for the cells to get rid of the old protein and make the new mutant protein. So it takes quite a long time of effort. This is why there's no such thing as a high throughput mammalian cell mutagenicity test. It's not possible to do this because you're expanding these cells to 10 to the 8th, 10 to the 9th cells. Meanwhile, you're looking for that 1 in a million in that population. So this is why there's a lot of chromosome and genotoxicity tests like the Comet assay that can be done in high throughput mode, but this kind of test cannot be. So what's the difference between DNA damage, genotoxicity, and mutations? Mutagens, when you expose a cell, they don't produce mutations. They produce DNA damage. The DNA damage is not a sequence change. It's basically something that's bound to the DNA. It could cause DNA strand breaks, but it's not a mutation. So you can detect, for instance, chromosome aberrations and damage. Underlying all this is DNA repair. So DNA repair happens and that damage gets fixed, you're not going to have a mutation. But since mutations are heritable, cells are the ones that make these mutations. They replicate the DNA, they go through a couple cell divisions. So mutagenesis itself is a cellular process, requires enzymes, causes DNA replication, it causes cells to grow, and more importantly, you're measuring this in a cell that's been damaged DNA and is able to survive that damage and pass it on. So it's not a dead end. It's a cell that can continue to grow. This is why genotoxicity is not equal to mutagenicity, because when you have a genotoxicity and you're measuring just that chromosome damage from that first round of cell division, you're not really measuring that transmission of that information to the next daughter cells. You're looking at a dead end. So genotoxicity is not equal to mutagenicity, and genotoxicity is required for mutagenicity to occur. So what are the types of mutations that we can measure in the laboratory? Well, the bacterial mutagenicity mammalian cell test, particularly like the Ames test, can detect point mutations. These are transitions and transversions. These are just terminology. Transitions is when you change one pyrimidine to another pyrimidine. And for instance, transversions, we change a pyrimidine to a purine, like guanine going to adenine. Guanine going to adenine would be a pyrimidine, a purine to a purine, and that would be a transition. Guanine going to a thymidine, that would be a transversion because you're going from a purine to a pyrimidine. The other type of mutations are base additions and deletions. These cause frame shifts. And it just means that you're in the DNA, you're adding a base, and now your frame shift, your triplet codon that is used to make proteins, now is haywire, so it's shifting its frame to maintain that triplet. So now, this particular one, you change a UCA to a CUC. And this can be an addition or a deletion. If you have a deletion, the same problem occurs. You now, again, change your reading frame from a UCA to a CAC. So these are frame shift and point mutations, base substitution, and these are the kind of mutations that are detected in the Ames test. The next slide shows chromosome mutations, and this is our large scale, and since bacteria don't have chromosomes, these don't occur in bacteria, you need a mammalian cell to measure these. And for instance, you can have very large losses, deletions shown here, where these are millions of base pairs of deletions. And unless you have an assay that can measure these, you're not going to see them. So for instance, you won't see chromosome deletions in a bacterial test. It just doesn't happen. It's not part of the biology. Some of these are really important, though. For instance, the one shown on the right here, this is the BCR-ABL fusion gene, and this was a break between chromosome 9 and 22. They got rearranged, and part of chromosome 9 got stuck in chromosome 22. And in this case, it forms a BCR-ABL gene, which is a very prevalent event in chronic myelogenous leukemia. So you can have a situation where you break a chromosome, and the two lesions come together and juxtapose next to each other some oncogenes that can now be very problematic to the cell. So what is the impact of these types of genetic damage? Well, there's two impacts. One is in our somatic cells, where you can have cancer, immune dysfunction when a cell that's mutated proliferates in vivo, or in the case of germ cells, you can produce mutant gametes, and that can result in transmissible genetic alterations. And this has been a large, the one on the right, germ cells, has been in the news a lot in science because there's a prevalence now of what's referred to as de novo mutations, where neither one of the parents has a mutation, but the child has a mutation. And some of these seem to be segregating with things like autism and some of the neurological diseases that occur in children. And that's a big research. What are the origins of these de novo mutations that are now being found in triplets of mother, father, and the child? But somatic versus germ cell is very, very hard to do. Germ cell mutagenesis is a very difficult problem to look at, and a lot of reasons has to do with the biology. A germ cell going from a primordial germ cell to a mature sperm takes in a human, takes about 70 days. In a mouse, about 6 weeks. So if you want to understand germ cell mutagenesis, this means you got to treat animals and measure it out here. You got to treat animals for a minimum of 6 weeks, 6 to 8 weeks. And so that becomes very problematic, very expensive, and very problematic because it's not something that is right now is required to do unless it's very obvious that there is an effect. This is also very different in the germ cells in females, because in females they undergo maturation very early on, and basically at birth the number of primary oocytes are fixed, and they progress to basically meiosis prophase 1, and they're kind of locked in that prophase 1 forever until puberty. And then when the cyclic effects of puberty causes these primary oocytes to undergo meiosis 1, meiosis 2, and form a germ cell, and then that is when this happens. So in the case of females, you have these cells been waiting around. So some folks are concerned about the continual exposure and the accumulation of things in the female, and it's a very different situation in the male, where they're constantly generating new germ cells in this continual process. So what are the consequences of mutation and chromosome anomalies? These include heritable genetic disorders, things like Fragile X syndrome, muscular dystrophy, Down syndrome, which is a trisomy of chromosome 21, but also somatic cell mutation can lead to cancer and aging. And cancer is the second leading cause of death in the U.S. So where does mutation fit into cancer? Well, over the many years, we've been able to isolate pathology lesions and progression from basically a normal cell to a tumor cell, and we've been able to isolate these and ask the question, what are the types of genetic events that are occurring during this progression from a normal epithelium to a colon cancer? And this is shown linearly here, but this is for this purpose. But you can see that there are certain types of genetic events in colon cancer that are associated with transitions from basically a normal epithelium to a small adenoma to a large adenoma, and finally things like p53 mutations, which we are believed to be that transition from converting from a large adenoma to a carcinoma. So these types of events have all been cataloged, and underlying all these are the whole world of epigenetic events, which is the subject of another discussion. Well, how is this information organized? Well, a decade ago, Hanahan and Weinberg came up with this idea of cancer hallmarks. And they came up with this framework where they asked the question, what are characteristics of tumor cells that make them a hallmark? And things like resisting cell death. If you have a tumor or a cell that does not die when you're treating it with chemotherapeutic agents, it doesn't die, escapes apoptosis, so it can continue to proliferate. We have a tumor that can induce angiogenesis, produce a blood supply. And so these are what we think of as the hallmarks, and these are types of events that occur that drive cancer. The other corollary to this is, can we use this kind of framework to design cancer chemotherapy drugs? So can we design, for instance, cancer chemotherapy drugs that can inhibit angiogenesis or cause cells to die or cause cells to proliferate or inhibit that? So we have a number of frameworks now for cancer hallmarks, and there's a lot of terminology out there regarding what these genes could be, what they look at. Some of these in the literature are called oncogenes, tumor suppressor genes, and oncogenes tend to be growth stimulating genes. Tumor suppressor genes tend to be inhibitors of cell division. So you're kind of looking at, for instance, analogy would be gas pedal versus the brake on a car. The oncogenes tend to put the gas pedal where they're driving proliferation, driving growth. And the tumor suppressor genes tend to put the brakes on the cells to inhibit cell proliferation, cause cell chocolate checkpoints, and also DNA repair. There's also the concept out there of driver versus passenger mutations, and this has come from a lot of the next generation sequencing where they sequence a tumor and they find 30,000 mutations in them. Well, not all of those mutations are contributing to the cancer phenotype. You have some mutations which we think are drivers, which are driving the cancer process. Others are referred to as passenger mutations, since they seem to be just going along for the ride, so to speak, and they're not driving the cancer process. They just be mutating as part of the process of that cell clonally expanding and continually to grow. So about a couple of decades ago, a fellow by the name of Bruce Ames came up with this paper showing that carcinogens are mutagens, and this kind of revolutionized the assessment of cancer. So you now had a potential to be able to assess cancer-causing potential in a rapid way. And in his case, he developed a bacterial cell system that enables the assessment of whether something can cause cancer within about 2 days, 3 days. And so this was a big revolution. And over the years, a number of papers have come out, and it basically has stayed pretty much online in that basically if you have compounds that are carcinogenic in 2 species, they're likely to be mutagenic in a short-term test, and they're also likely to be potential human carcinogens. So there is a basis for using short-term tests as a basis to predict potential cancer risk. There's also the International Agency for Research on Cancer, and they have what's referred to as the Group 1 human carcinogens. And these are compounds that are carcinogenic in at least 2 species. They're genotoxic in mutation tests, and they induce tumors in multiple sites at multiple sites in these animals. So you have these types of compounds where we cause tumors, mice and rats, they're genotoxic. Those compounds tend to be potential human carcinogens. So what is the key events for a mutation? Well, you have to have DNA changes that result in a mutation. It has to be able to do that by a metabolite or indirectly, and this is considered a cancer key event in compound-induced or xenobiotic or foreign chemical-induced cancers. And the US EPA guidelines consider this kind of early tumor response initiator, and all these are types of events where we can fall under the category of a genotoxic mode of action. Although we are exposed to these compounds continually, we have Mother Nature has designed a number of DNA repair systems that keep us healthy. And this is very important because if you have DNA damage through either an endogenous or exogenous exposure, if it's unrepaired, you can have a cell that begins to accumulate DNA damage. This cell can either go into senescence, basically stop growing, can go to become apoptotic. However, if it accumulates a sufficient amount of DNA damage in those target genes, some of those cancer hallmark genes, this can contribute to cancer and aging. So the potential consequences of unrepaired damage are you can have chromosomal changes, which affect thousands and millions of base pairs, structural changes, or you can have gene mutations that affect very finite number of bases, 1 or 2 or 3. And more important, as importantly, is this whole category of numerical changes, aneuploidy. We know aneuploidy is really important for cancer, and having systems that can detect that is very important. As I mentioned earlier, we have a number of lesions in our DNA, but for almost every single lesion you can imagine in your DNA that's been found, there is a repair system that is designed to protect that. Protect against it. For instance, bulky adducts here, we have nucleotide excision repair that enables the removal of that adduct, this thymidine adduct, and be able to form back be error-free. We also have a number of sites which reverse certain lesions, alkylating agents, O6-ethylguanine, which basically is a complete reversal. Some cross-links are more problematic for cells because when you're replicating DNA, if you have a cross-link, you can't unwind the chromosomes, and that can be a very lethal event to cells, and that can cause chromosome breaks, and there are a whole number of systems to repair these. One of these is called non-homologous end joining. In that case, by the very definition of it, it's a mutagenic event. It's error-prone system because it's non-homologous end joining. It's putting two pieces of DNA together that don't belong together, but it's forming that so the cell can survive. So nucleotide excision repair is one of the more common repair pathways. This repair pathway is deficient in the bacterial mutation assays, which enables and increases the sensitivity of the assay. And this is just a cartoon of that, where basically a lesion is recognized, a thymine dimer is recognized by enzymes that cut it out, cut that lesion out. And since there is a template on the top here, you can reform that synthesis using the other template as a guide, and you can reform the whole DNA molecule, and it's error-free, typically error-free. So that is an error-free repair system, and as I mentioned, it's deficient in the Ames test bacteria, which enables the detection of compounds that cause this because you can't repair them. Now, excision repair is really important for cell survival, not only to UV radiation, but a number of types of exposures. And this shows cell survival in the wild type UVR+, which is the excision repair system, versus a UVR- cell. And you can tell very quickly there that if you have DNA damage and you don't have that repair system, it's deadly to the cell. It will kill these cells. These are log order magnitudes of cell killing. So it's a very, very important system to have to protect against UV light, also against other chemicals that may cause DNA damage. This is also important for human cancers. There are a number of human cancers that are heritable cancers that have a DNA repair deficiency. One of the most common ones that you hear about, for instance, is xeroderma pigmentosum. And these are children that can't go outside because of the sun. They can get skin tumors. They are not typically go outside. If they go outside, they'd be very heavily protected. A number of other diseases are out there that are associated with a DNA repair deficiency is, for instance, BRCA1, BCR2. This is a very uncommon mutation in sporadically. However, if it's a familial mutation within a family, it becomes very problematic, and it's a very high frequency event of developing a breast cancer. But in the general population, it's not really that prevalent of a mutation. It's as common as some of the other ones. So, however, within a family, it's critically important. So over the last 10 years or so, Genotox test battery, the original test battery, was published in 1997. And this was a test battery to be able to detect both gene mutations and chromosome aberrations in a fairly rapid sense and be able to predict the potential to cause cancer. There are two options for the test battery. Option one, this is from the ICH, which is the pharmaceutical industry genetic toxicology test battery. One is a gene mutation for bacteria to detect basically gene mutations or point mutations. The second test is a cytogenetic test to detect chromosomal aberrations. And the third test is to detect chromosomal damage in vivo. The second test there are all in vitro tests that are used to detect chromosomal damage in vitro. Now, there have been some revisions to the genetic toxicology test guidelines. I was on the expert groups that revised these. They've all been revised. The guidance is shown here, OECD guidance, with the exception of the very first one, OECD 471. All the other ones were revised in 2014. One of the big changes that you should be aware of is the addition of OECD 490, which just happened recently. This replaces 476, and as I mentioned down here, 476 is now HPRT XPRT mutation. OECD 490 covers the mouse lymphoma test and human TK6L gene mutation assay. The option 2 is basically used as well, but it's not as prevalent. Most folks do option 1 plus option 2 basically so that they don't get questioned. And option 2, the big difference is the incorporation of the Comet assay. And in this particular case, the Comet assay is recommended to be done in the liver and liver only. At ILS, we have ability to conduct a combined micronucleus and Comet assay, so you can do both assays in one study and get both endpoints measured in the same study and fulfill requirements for both OECD 489 and 474 in one study. Well, when you look at all the data, you ask the question, how good are these at predicting cancer? Well, they are pretty well. So we know that these are the group 1 carcinogens, human carcinogens, and either use a gene mutation assay or a chromosome aberration assay, you can detect the activity of all these human carcinogens. So this gives idea as to why we use this test battery. The EPA also has a test battery for new chemicals, and it's shown here. One of the important differences in EPA is there's exposure-based testing. So if you have human exposure, they want to test it. And this first component shown here, which is SAR. So this is structure-activity relationships that are done. These are computerized programs that allow us to determine whether there is a potential mutagenic risk or genotoxic hazard with this chemical, and that would trigger testing. And so those are very important. But the remaining test battery is basically identical to what you can see for the pharmaceutical industry. So as I mentioned, the ICH guidelines were revised in 2011 from 1997. And this was because there's been sufficient data accumulating over time to show that the mammalian tests that are used have a very high false positive rate. This was very problematic where the false positive rate was basically unacceptable. And there were a number of workshops that were convened to be able to ask the question, why are these cell types and these cells in this test battery, this particular ones, in vitro ones, why do they have such a high false positive rate? Well, when they did some of the genetic testing on these, they discovered that the mouse lymphoma cell, CHO cells, and some of these rodent cells, they all have p53 mutations in them. The mouse lymphoma cells have p53 mutations in both alleles of the p53 gene. This means that their DNA damage response is compromised, which could lead to, when you do testing, if you have a compromised DNA damage response, this could lead to false positives because chemicals that would normally be repaired or fixed by p53 are not, and they're going on and forming DNA damage. This is in contrast to human TK6 cells, which are wild type for p53. They're a human cell, they're wild type for p53, they have an intact p53 DNA damage response, and those are the cells that we use at ILS. We use those cell lines, this cell line, human TK6, for our in vitro micronucleus assay because it's a human cell line and it's p3 proficient, which we think is very important to prevent false positives. And we want to prevent both false positives and also false negatives. So we want to protect against those, and one way to do this is to use a human cell line that is p3 proficient. As I mentioned, the genetic toxicology test lines guidelines have all been revised except the Ames test. These are all revised in 2014. This one here, 490, was revised in 2015. But since all these were done in 2014, they are now in effect. After an OECD guideline gets approved, finally, it's about a year and a half afterwards where it actually takes effect. So these now done in 2014 are taking in effect in 2016. So going forward, you have to comply with all the revisions to these guidelines that were done over the last couple years. One of the areas that I mentioned for is structure-activity relationships, and this was originally conceived by a fellow by the name of John Ashby, where he produced this chemical structure, which is a fictitious chemical structure, but it has every structural alert that we can think of that can contribute to mutagenicity. So you have things like an epoxide here, which we know is electrophile that can bind to DNA. You have other leaving groups here, which can form. So this idea of can we come up with structures and be able to decide whether that compound has a potential risk was pioneered by the fellow by the name of Herbert Rosencrantz at Case Western University. This is why it's called multi-case. And he has developed a number of programs, and since then there have been a number of programs that have been developed that allow structure-activity relationship assessment and allows you to decide whether you should proceed with a genotoxic test or not. And right now, usually, for instance, the FDA and EPA require 2 programs to be run to come up with your idea of what is it potential hazards of this chemical, purely based on structure. So for now, I'll go through the other genetic toxicology tests that are required. And the first one that I'll describe is the Ames test. This is a bacterial test that's been used for decades to assess genotoxicity. It's referred to as the Ames test because of Bruce Ames, but Bruce Ames really only designed the salmonella assays. Later on, there were some E. coli tests that were part of it that were developed by a fellow by the name of Green. But this is a test which uses bacteria which require histidine to grow, and when they become mutated, they don't require histidine to grow anymore. So you're able to do this very easily. You mix the cells with a liver enzyme prep, which is referred to as S9, which I'll talk about later, which is a source of cytochrome P450 enzymes and enzymes present in the liver that metabolize compounds. So you mix these 3 together, and your test agent, you mix them together, pour them on a plate, and then you incubate that for 37 degrees for 2 to 3 days, and then you count the colonies on a plate. And these are colonies now that have gone of cells that have gone from histidine requiring to not requiring histidine, and these are referred to as revertants. The genotype of these strains is shown here. These are the strains that were used in the Ames test. Typically, the OECD guideline 471 requires 5 strains to be used. This shows the strain, the genotype, and some of the other genetic variants that are built into these strains. For instance, as I mentioned, they are deficient in DNA repair, UVRB, they're deficient in that. And some of these have a plasmid called PKM101, which carries an error-prone DNA repair system. The strains that are typically used, for instance, at ILS are TA98, TA100, 1535, and TA97A, which is a version of this TA97 strain. One thing you might note is that these strains, the ones that I just mentioned, all have GC base pairs at their target site. So someone decided we need a strain that's got AT base pairs at its target site. Well, a salmonella strain was developed, but it became very problematic because it had a very high background, it was unstable, and this is why these E. coli strains came in now. A fellow came up with these E. coli strains which are exactly the same, they work exactly the same, except they require tryptophan instead of histidine, it's an E. coli strain, but you now have a situation where you have an AT base pair at that target site instead of exclusively GC. So when you do these combined strains, these 4 tester strains, T98, T100, 1535, and 97, as used at ILS, in an E. coli strain, you now have covered both GC and AT base pairs with these 5 tester strains, and allows you to detect a very broad range of mutational types. The next tests are chromosome aberration tests. Again, as I mentioned earlier, you think about the cell cycle and the time it takes for cell cycling. So there are 2 versions of this. One is using human lymphocytes, blood samples, or again, Chinese hamster ovary cells. And basically, you do the exposures, and you incubate the cells for 1.5 cell cycles to be able to catch them at the metaphase chromosomes. And then you harvest the chromosomes and then the cells, and then someone counts them and looks at them on a scope and counts them individually. As I mentioned, right now the criteria is 300 metaphases per data point per tissue or cell sample. So it's quite a bit of time and effort, and takes certain expertise to be able to count these chromosomes in these cells. However, a few years back, it was discovered that instead of counting chromosomes and looking at this, someone discovered if you let the cells go on to an extra division, that chromosome material that was broken off, chunked, gets formed into a micronucleus. And this discovery made scoring chromosome aberrations very easier, much quicker, faster, and also ability to automate, which you couldn't do before. So the micronucleus shown there is actually a fragment of a chromosome that was broken off, or an entire chromosome that when the cell went through division, it passed it on to the daughter cells, and that micronucleus is capturing that chromosome piece that was broken off, or the entire chromosome that was fractured off the cells or taken from the cells. And this is shown how you form micronuclei. You can have a break, which forms a break. The cell now divides. They have the 2 large nucleus, and you have a little micronucleus shown here. If you have aneuploidy, and this is becoming actually very important to be able to measure aneuploidy, because none of the tests that we have, it's very difficult to measure aneuploidy. And in the case of micronuclei, we can score micronuclei with both breaks and aneuploidy. The aneuploidy micronuclei tend to be a larger size, and there is actually some immunohistochemical stains that can be used to make sure that that is an entire chromosome. For instance, you can stain for the kinetochore of the chromosome, and that will tell you that the kinetochore is in there, and therefore you have an entire chromosome instead of just DNA. However, in our case at ILS, we don't score by microscopy. We use a flow cytometry-based assay, and that's shown here. It was developed by Litron. It's a dual staining assay, which allows you to stain the cells and identify the necrotic apoptotic cells from the living cells, and then you can score the micronuclei, and you can also score relative survival by using a bead-based assay. So this can all be done by flow cytometry. And on the next slide shows what our assay looks like here at ILS, where we use human TK6 cells. Cells are exposed with and without liver homogenate for 4 hours, or 24 hours without any liver homogenate. The cells are incubated. We let the cells divide through 1 cell cycle to capture the daughter cells, and then we can use a flow cytometer, and that flow cytometer captures relative percent survival, percent apoptosis and necrotic. It can also tell you the cell cycle phase, and gives you the micronucleus frequency. One of the problems with this assay has been this point here where the survival needs to be 55 plus or minus 5%. And that's part of the OECD requirement, and that becomes a pain point, because if you miss that window, that 10% window, you can say that your test was not valid. And it's not a problem if you go above that toxicity if your compound is negative. But if your compound comes out positive and you exceeded the toxicity limits, your client won't be too happy with you, because you've exceeded the toxicity limits, you got a positive, and you're going, well, it's not a valid test. But if it's negative, it doesn't become an issue, because you've tested the limit of the assay, you've tested cytotoxic concentrations, and you know that that compound was tested appropriately. The in vivo micronucleus assay is the in vivo version of a chromosome aberration test. This is an assay that uses animals, requires either single treatments. If you use single treatment of animal, you need to look 24 to 48 hours afterward because we're scoring typically in the peripheral blood. The other approach is to treat the animals every day, or 2 or 3 days or more, and then collect the sample the next day. The top dose in this study with OEC 474 is 2,000 mg per kg. It's pretty massive doses, unless there's data showing that lower doses are lethal. And again, you can either prepare cells, stain them, and count them on a microscope, or you can use a flow cytometer. And at ILS, we use, again, a flow cytometer to do these assays. And why do we wait 2 or 3 days? So as I mentioned, they dose the animals and you wait a few days. This is because what we're measuring is DNA damage that occurred in the bone marrow of the cell, of the animal, and that cell has got to mature and go into enough maturation. For instance, we know red blood cells don't have nucleus. Well, the erythroblasts do have nucleus that are in the bone marrow, and as they mature, the main nucleus is extruded out of the cell, and basically you form a cell without a nucleus, which is called a reticulocyte, which is an immature red blood cell, and through further maturation, you get a mature red blood cell. If you have a DNA damage here, you go through the cell replication, and as the mother nucleus or the large nucleus gets extruded from the cell, the micronucleus stays behind. And you can actually measure this. So you have basically a cell with no nucleus, but has a micronucleus in it. You can either do this by the reticulocytes, which are CD71 positive cells, or we can let them go on further and look at the mature red blood cells. And then mature blood cells that have DNA in them, a small chunk of DNA, are micronucleated red blood cells. These micronucleated red blood cells are measures of chromosome breaks that occurred, or aneuploidy that occurred in the bone marrow that we measure in the blood. Increased levels of chromosome aberrations and micronuclei have been associated with cancer risk. It's not a huge cancer risk, but for instance, in European groups, they're able to go back to old blood samples and ask the question, these people now are in their 60s, what is the cancer risk? And it turns out people who have elevated aberrations early in life, they tend to have an increased cancer risk. It's not astronomical, but it is an increase. The other important part of this biomarker is it can be done in mouse, rats, dogs, and in humans. So you have a potential to go into any species on any study and be able to ask the question, is there chromosomal damage? And you can actually add this to 14-day, 90-day studies. We do this for the National Toxicology Program, where we look at 90-day studies and measure micronucleus in those studies. The other study is the Comet assay. The most recent guideline is Guideline 489. This is a new guideline, not a revised. This is for the Comet assay. And the Comet assay, just a little bit more detailed because it's a newer assay. ILS has a lot of expertise in this assay. It has been done for decades. Is the single-cell gel electrophoresis assay. It detects a broad range of damages. It has an advantage in that you can look at any cell in any tissue in any time in the animal. You have to be able to make a single-cell suspension, which is basically easy to do for most tissues. Some of the disadvantages of the assay, it's very slow. It requires microscopy scoring. You've got to have harmonization amongst the staff. Cell necrosis, apoptosis is a potential confounder. And again, in the Comet assay, you're measuring DNA damage. You're not measuring the transmission of that DNA damage to daughter cells and those cells living and causing a mutation. You're just measuring the initial DNA damage. There was a JAK-FAM validation trial that was ongoing to validate the in vivo Comet assay. ILS was one of 2 laboratories in the United States that participated in this validation. It was led here by Cheryl Hobbs. And this OECD guideline was approved in 2014, published, and now it is in effect. So the principle of the Comet assay is either you have cells exposed or animals exposed. You make a single cell suspension from that tissue, liver, for instance, or a cell culture system. You mix that with low melting point agarose, and you put it on a slide. You lyse the cells basically in situ on the slide, and what you end up with is little nuclei or balls of DNA that were the nuclei that were left behind in the cell, embedded in the agarose. And then you do electrophoresis on that, and you form basically a comet, and that's what basically is the nuclei of a cell electrophoresis. And as you increase and so these are quantified using image analysis systems, and for instance, ILS has a Part 11 compliant image analysis system which enables us to capture these images. And the metric that's being used right now is this percent tail DNA, which is a calculation of how much DNA is present in the tail compared to what's in the head. And the software, what it does is it captures this image and quantifies the DNA that's in the nucleus, what's in the cytoplasm, and then has, for instance, like these bars here, which are background correction, so it subtracts out the background. And this type of system is used to be able to quantify these. And this shows the progression of DNA damage. As we analyze comets, you can see that the olive tail moment, or percent tail DNA, goes up with increasing amount of DNA damage. And even here we have a cell that has a very heavily damaged cell. This is something that can be scored still. And therein lies the problem, and this is a big, huge debate, is do you score these at the very bottom here, or do you not score them? There is a terminology in the literature referred to as called hedgehogs, and hedgehogs are believed to be kind of a cytotoxic measure in this assay. And so when we do this assay, we not only score for these cells, but we're also looking for heavily damaged cells, which are referred to as hedgehogs. In this particular case, this is a scoreable cell. It's not considered to be a hedgehog, but it's considered to be a heavily damaged cell. And it's still part of the calculation. So you can see, if you have a lot of these, you're really going to sway your data. So you got to be careful when you do this assay. And also, capturing these images, deciding which image to capture and score is a problem as well. And we solve that by harmonizing our scores. Our scores are harmonized every year to make sure that they're seeing the same thing and scoring the same thing pretty much the same way. Just a little bit about the guideline. It was established in 2014, and one of the points about the guideline is we have to score 150 cells per tissue. In the past, we only scored 100 cells per tissue. That has gone up to 150, making it more time-consuming. And then also there's a quirk about analyzing the data, where basically you're using the median of each animal, and then you take those medians and you calculate a mean. So you actually report the mean of medians per group, which is a quirk of how you do the calculations. So how do you interpret these data? Well, the way the data are interpreted is you have to define what are clear positives or clear negatives. So as part of the OECD expert working group, we had to deal with these cross-cutting issues. So what would be positive? So we had to come up with a criteria that we can use to judge all the data. So these are the three criteria that you use for any Genotox test battery for each test, and you make the decision whether something positive or something is negative. At least one concentration has to be statistically significant. You have to have a dose-related response for at least one dose. And your results have to exceed your 95% confidence limits of your historical control. These three criteria, if they're all met, you have a positive compound. If none of those are met, they're all negative. If one of these or two of these are met, you have what's referred to as an equivocal compound, and that's when you exercise your scientific judgment. And so when you have these type of criteria, where one or two, you can put in equivocal, but then you got to assess the relevance. So in our case, when we see something that is statistically significant for that particular day of the test, you have a dose response, but it does not exceed our historical control, we don't think that may be as biologically relevant as something that meets all these three criteria. So the language in that and imposing our scientific expertise and judgment on this becomes very important, because as you likely could appreciate, this happens a lot. We have compounds and we test where we have two of these met and not all three are met, and we have to make a scientific judgment call on what we think the data is telling us. So we have these type of positive tests. What can you do to follow up? I just want to remind people that the Ames test is DNA repair deficient, makes it very sensitive. Most cells are DNA repair proficient. So you have a tester strain that is unusually sensitive to DNA damage because it can't repair anything. There's also the issue of xenozoic transformation, where you have bioactivation and detoxification. The in vitro assays, we use a liver homogeneous preparation to be able to metabolize compounds or biotransform them to more reactive metabolites. This is an enzyme system that's been induced, so you induce the liver of an animal. However, that's not how drug metabolism works in vivo. This is what we refer to as phase 1 metabolism, P450. It doesn't have phase 2 metabolism, and that's critically important because both in vivo, the phase 1 metabolism and phase 2 metabolism are linked. Phase 1 metabolism tends to generate or create functional groups that are substrates for phase 2 metabolism. And when you have these two disconnected, it can result in a lot of bioactivation in the absence of detoxification. And these are phase 2 enzymes that require cofactors and that are not added into the S9, but are active in vivo. So when you transition from an in vitro system for S9 versus an in vivo system, you have two very different enzyme systems that are being used to metabolize that drug. And the in vivo situation has both. The in vitro system is basically geared towards detection, not so much being able to show the detoxification of these compounds that we are exposed to. For far as follow-up tests, there's a transgenic mouse mutation assay. This is, again, OECD 488. This is a 28-day exposure with the 3 days and there's at least 3 or 4 models that meet this, big blue mice and rats. I spent quite a bit of my career on early on, the Mutamouse. But more recently, those transgenic mouse mutation assays require a specific animal, and animal models, they're not really wild-type animals. They're animals that have been used traditionally in toxicology. So there was a need to develop an in vivo assay for mutation to complement the chromosome test that can be used on any animal. And this is the flow cytometry PIG-A gene mutation assay, which is being developed by Lutron and a couple other groups, which allows us to use a cell surface marker on a red blood cell to assess mutation. So when that marker is lost, we have mutation because that enables us now to be able to detect that on a flow cytometer. So this can be done by flow cytometry, automated, and can be used to detect mutation at the PIG-A gene. There are also a number of screening assays that, for instance, ILS has been developing here and other places have developed these. One is the mini Ames test, and these are basically the same strains that we use in the traditional tests. However, we scale it down into 6-well plates or 12-well plates, and this typically uses 2 or 3 bacterial strains that are used in the Ames test. This saves compound time because the Ames test uses 100-millimeter dishes and very big plates. To do this test requires more compound, and these tests require less compound. They're not any faster, but require less compound. The Ames 2 is a liquid format run test, and this is a 384-well format test, and you're not measuring colonies, so you're not really duplicating what you measure in the Ames test. These mini Ames typically use TA98 and TA100, and those tend to capture about 90% of the positives you have in these tests. So this is why you can use those 2 or 3 strains in that test. It's very useful for pharmaceutical companies that may have a number of candidates that they want to test before they go into drug development or further tests. So you can screen these 10 or 12 compounds and identify the bad actors up front and allow you to run your Ames test on what you think is going to be a compound that will not respond and that allow you to go further in drug development. The other is the green screen. This is an assay that uses TK6 cells, and it's using the GAD45 gene linked to a green fluorescent protein. So when you activate the DNA damage response, the green fluorescent protein gets turned on, and again, you can measure this on a flow cytometer. The Tox21 efforts, there's a lot of high throughput efforts ongoing right now by the EPA, FDA, and one of the outcomes of that has been a particular assay using H2AX, which is a reporter assay which detects DNA strand breaks. And finally, one of the efforts that ILS has been heavily involved in is using genomic expression profiling to assess genotoxic versus non-genotoxic modes of action. And this is the concept of identifying gene signatures that are indicative of a genotoxic response in cells or a non-genotoxic response. So you can identify potential bad actors by using gene expression profiling. This can become high throughput in nature over years of the next coming years using next generation sequencing technology, and can be applied either in hepatocytes in culture, human TK6 cells, and rodent liver. And that paper Yock et al. Is where we use this technology in a collaboration with Health Canada to be able to identify a gene signature set which can identify potential genotoxic compounds using human TK6 cells, and not only identify them, but identify their mode of action. So when we have a genotoxic response in the cell system, do we think that was due to oxidative stress? Was it due to aneuploidy? Was it due to DNA damage? And by using expression profiling, you can sort that out. Finally, the last slide is can we do all these measurements in humans? And the answer is yes, we can. And there are a number of papers out there where they've gone into people and measured things like DNA adducts, ethylene oxide in humans. That was one of the data sets that was really important in making ethylene oxide a human carcinogen. The revelation that we have people around that have ethylene oxide DNA adducts, chromosome breaks. You can measure the Comet assay. As I mentioned, the micronucleus assay can be done in a number of species, including humans. The HPRT assay has been done in humans as well, using T lymphocytes. And finally, the PIG-A assay can be used to measure mutations in humans as well. So there's a whole lot of ability to go directly into humans and measure these kind of events in humans. This is a little concerning to a number of folks because what are the health consequences of a measurement of a chromosome aberration in a human or a micronucleus? We're uncertain, but we think that may be an important measure to do, and there's been some talk about incorporating some of these into phase 1 and phase 2 and phase 3 clinical trials. So with that, I'll end and be happy to answer any questions.